All right. Hi, everyone. My name is Grace Kirkpatrick. I've been studying biochemistry and molecular biology in the College of Arts and Sciences. And for the past year, I've been working in the Laboratory of Molecular Neurobiology under the supervision of Dr. David Farb and Dr. Vidya Kumarasan. My project that I will be presenting to you today is about Alzheimer's disease and correlating behavioral deficits in learning and memory to neuropathological changes in the hippocampus. So before I talk about my project, I want to give you guys some background knowledge on Alzheimer's disease. Alzheimer's disease, shortened to AD, is a neurodegenerative disease that affects 40 million people globally. In fact, in 2014, healthcare costs were estimated to be around $172 billion, and according to the Alzheimer's Association, this is expected to rise to $290 billion this year. Disease progression is characterized by severe cognitive deficits, such as dementia and memory impairment. These behavioral changes can be traced through pathological or physical changes within the brain that are related to this disease. For example, here we see an image of the brain on the right side. On the left side is an image of the brain that's very healthy looking tissue, whereas on the right side you see a brain that's severely atrophied, one that you'd expect to see in a patient with severe Alzheimer's disease. Now, one area of the brain that's especially affected is called the hippocampus, circled in turquoise, and this is the part of the brain that I'll be focusing on for my project. So the relationship between Alzheimer's disease and the hippocampus. So the hippocampus is this part of the brain in red, and it is important for spatial awareness and memory. So this is the part of the brain that you use when you walk into a room. You're able to orient yourself, you know, where like the chairs, the desks, the windows are, and you are able to store this information and regurgitate it at a later time. Now, people with AD experience extreme neuronal loss or cell death within this region of the brain. This in turn leads to many of the cognitive deficits we see, in particular memory loss. So here I have a picture of like the cross-section of the hippocampus, and this is what we are looking at when we're analyzing these like changes. The cells are outlined in blue. And a fun little fact, the name hippocampus comes from the curved nature of this region of the brain that resembles a seahorse, which is part of the genus Hippocampus. All right, so then some additional hallmarks of AD within the brain include amyloid beta plaques, and these are pictured on the right, and they're the big brown blobs. Another standard hallmark are neurofibrillary tangles, or NFTs, which are comprised of this protein phosphorylated tau. Now, why is this important? So tau itself is important for cell stability, is highly prevalent in areas of the brain such as the hippocampus. But phosphorylated tau is actually very detrimental and leads to a lot of neuronal instability as well as cell death. So my project is looked at phosphorylated tau and is looking to correlate behavioral changes that occur in the hippocampus that are caused by the hippocampus in the presence of this protein phosphorylated tau. So for the project, we have two types of rats. The first type is the wild type, which is normal and healthy, and we're comparing this to this transgenic or Alzheimer's diseased rat. The idea with this Alzheimer's rat is that we're basically expressing genes from human Alzheimer's disease patients so that they basically mimic the symptoms and the signs that we would see in humans. So my project that I worked on is part of a much greater project. And this basically has two parts. The first part is the behavior, which we used these novel object location tests to basically correlate changes that we see in the hippocampus. And I'll go into a little bit more detail later. The second part is the neuropathology, in which we use staining using immunofluorescence to look at the protein markers within the brain. And the idea is that we're testing rats at 6, 9, 12, and 16 months so that we're able to see this gradation of changes throughout AD progression. And then basically can correlate the changes that we see with the protein as well as the behavior. So to break down what happens with a novel object location test, the idea is that we first put a rat in a training environment for about 10 minutes with 2 identical objects in a set location, and we allow them to explore. Then we remove them for 20 minutes, and then put them in a new location, or a new environment with 2 of the same objects, but 1 moved into a novel location, and allow them to explore for 5 minutes. We then record this, as shown here, and are able to then basically track the interaction time that the rat has with these certain objects. Videos are then compiled and then analyzed using Cineplex Behavior Research System so that we can tabulate interaction times, which give us a very large data set that we can then analyze statistically. So with the data I collected over the last semester, we basically tested 19 rats of varying ages, 6, 9, 12, and 16 months, all of which were male. We basically tested their interaction times at the 2 different locations, as shown on the bottom of the graph, the familiar location and the novel location. The wild-type rats are in pink, and the transgenic are in green. And so the first test I ran was a Kruskal-Wallis test, which basically would allow me to see if there's a greater chance that one of the groups has a greater chance of having a higher time of interaction than the others. So once I ran this test, I obtained a significant result, which basically means that yes, there is one group that has a higher chance of being having a greater time of interaction than the others. However, I did not know which group this was. So then I ran another test, the Mann-Whitney-U test, and comparing the values at the novel location between the wild type and the transgenic. After running this test, I also obtained a significant result, which means that there is a significant difference in the amount of time of interaction between these 2 groups. In particular, we see that the transgenic spends a lot less time at the novel object than the wild type. And this is especially exciting as it shows a decrease in hippocampal function. This is explained because rats are exploratory by nature, and so we would expect a healthy rat to actually spend more time at the novel object like we see in the wild type. This behavior can be related to humans because imagine you're walking down like Comm Ave and you see, things that you normally see, the bus, you see people walking, you see bicyclists. You don't spend a lot of time like looking at these things because they're very familiar. Now compare this to when you're walking down and you see a Zinneken's food truck, which you don't see all the time. This is like a novel object in like a new location, and you spend a little bit more time like observing that and the things that you do on a day-to-day. So this result is really exciting because it means that we can go into the next part of my project on the immunofluorescence, knowing that we're seeing these behavioral changes and we can correlate them to differences in the protein that we see in sections of tissue. So for example, immunofluorescence, if you forgot, is basically a fancy word for staining. This is an example image of staining in the hippocampus, looking for the protein V-GLUT. So for my project, I primarily focused on detecting phosphorylated tau within the hippocampus, a protein shown here. We basically have 2 different flavors that we can use to detect this, AT8 and AT180, and the only difference is that they're both detecting different types of this phosphorylated tau. For those of you who are unfamiliar with the process of immunofluorescence, the idea is that you basically take these free-floating sections of tissue, put them in a 24 microwell plate shown here, and expose them to a series of washes to prepare them for further staining. With the primary antibody, you can detect the protein of interest, like phosphorylated tau, and you usually do this overnight at 40 degrees Celsius, so then you can then add a secondary antibody, which will then let you visualize this protein. And then you get these really beautiful images as shown here. In the red, you see is detection of the phosphorylated tau. In blue is the cell bodies, or inner cell bodies, and this was taken in this region of the brain called the CA1. Here is another image of staining in a different region of the brain, the CA3, and again we see red shows phosphorylated tau, and then the blue showing the inner parts of the cell body. This is compared to the control, which we did, and this is where we did not add a primary antibody, which means that we would not expect to see any staining. And so because we see limited to no red staining, we can basically assume that any of the staining we received in the last section, the last 2 sections I showed, is indeed real staining. And so one of the big things that we're going to be working on in the lab is to continue optimizing the staining with with antibodies AT8 and AT180. Some of the slides that we used were not as sufficiently stained as we would have liked. And so we're going to be continuing to work on making sure that our stains are as successful as possible. Another aspect of this is starting to analyze results between different rat types, wild type and transgenic, as well as at different ages, 6, 9, and 12, and 16 months. We're going to continue collecting data from behavior analyses with rats at different age groups, 6, 9, and 12, and 16 months, so that we can then further correlate the differences that we see across both the behavior and the immunofluorescent data. And then finally, we're also going to be looking at additional targets such as GAD67, V-GLUT, and parvalbumin, so that we can get a clear picture of AD progression within the hippocampus. At this time, I'd like to give some thanks to everyone in the FARB Laboratory of Molecular Neurobiology, Dr. Tate Farb, as well as Dr. Vidya Kamarison. Thank you so much for your support throughout this project. Thank you to Sam Cook for your statistical analysis expertise, and also to my mother for always being my sounding board and being there for me throughout this entire process. Thank you, and are there any questions?
Are there reasons why you chose a rat, not like a mouse or any other?
Yeah, so with rats, the model that we actually chose came out about 5 years ago, so it's fairly new. And the thing is about rats is that they are actually much closer in evolution to humans than mice. And the model that we particularly chose shows a lot of these very specific pathological factors that we see in the brain that some mice models did not. So, yeah.
Why did you, for future testing, why did you choose those age ranges?
Right. So I personally did not choose the tests, but the idea is at 6 months, we're not really expecting to see a lot of changes. However, by 12 and 16 months, it's you're nearing the end of a typical lifespan of a rat. And so you're gonna be seeing those really severely affected Alzheimer's disease sort of symptoms. And it's basically evenly spaced between. Yeah. Thank you all.